For this flower dissection, we are using hibiscus flower. These are the brightly colored petals and these are green colored sepals. Firstly, with the help of a blade or needle, we will make a small incision so that sepals can be separated easily. Make sure you do not make a deep incision, otherwise the inner floral parts may get disturbed. Let's keep it aside. Now, we will remove all the petals one by one. Petals are generally brightly colored to attract the pollinators. As we remove these petals, we can see the reproductive parts further clearly. These are the petals. We can now slit open this filament with the help of a needle or blade. The slit should be made till the tip. We should make the incision very carefully as there is style within this filament. Now we are going to go ahead and separate the pistil from the stamen. We must pull this out gently so that both style and stigma gets separated from the stamen. Here it is. This is the pistil and this is stamen. Let's separate both pistil and stamen from the flower. This is pistil. You can see this is stigma and style. This is anther and filament. If we remove this filament, we can further dissect and see the ovary at the base. Let's remove this part. We can make a slight slit and remove the leftover filament. As you can see, the ovary is visible now. This is the ovary, and if we make a fine incision, we can see the small ovules which are present inside. The small dot-like structure which are visible, they are the ovules. So this is the pedicel, ovary and ovule within. These are the parts of the flower.